You're welcome once again. This is, I think this should be the last class that I have with you. So I will just quickly revise. I will just quickly do this topic before we start our revision. So you're welcome to CRP305. I was very, very busy in a workshop. We are on till like past 3. So that's why we couldn't have our work in the morning. Okay, without wasting time, let's quickly talk about hybridization. And that is just simply the creation of hybrids. Hybridization is the creation of hybrids. And how do we do that? It's first of all a method of crossing. Here you create hybrids, and hybrids are just new, they are kind of new individuals, okay? And you do that by crossing 2 pure lines. By now you should know what a pure line is. So they are pure lines, but they don't have the same genotype. So we have a pure line that has the TT genotype, capital T, another pure line that has a small t genotype, and you cross it together, you're going to get a hybrid, F1, which is capital T and small t. So the success or failure of a hybridization program is determined largely by the choice of your parents. And like I said, you are crossbreeding genetically dissimilar parents to produce a hybrid. So what happens to the genotype when you continue to self? Okay, you know, when you get your F1, you have to self it to get F2, you self to get F3. So when you continue to self, what happens? The first thing that happens is that heterozygosity will decrease, okay, because it is tending towards one genotype. So heterozygosity decreases while the homozygosity, it increases. The genotype is tending towards is the one that's increasing. So for each generation of selfing, you will lose half heterozygosity to homozygosity. So homozygosity is the one that increases while the heterozygosity decreases. That is what happens when you self continuously. And we briefly want to talk about the pedigree method of breeding. Pedigree simply means the family history, the history of a family. It also happens in crops. If you want to know the history of this particular crop, you have to go to the pedigree, or you have to perform what is called pedigree form of breeding, because sometimes you have to go and look at the descendant of that. That's for breeding purposes. You look at the descendant of that plant to be able to know which kind of traits you want to work upon, the one you like there. So pedigree breeding consists of crossing parents and generating segregating populations, okay? And you normally do that in self-pollination crops. I'm just trying to fulfill all righteousness with this pedigree method of breeding. I don't really think I have any question on it, but I want you to know what it means. So it is a method of breeding that you also use. Breeders use it to develop new varieties in self-pollinating crops after hybridization. So what they do is that after forming hybrids, you now want to go and like kind of form a family tree, so that anytime any breeder wants to know about this crop, you can always go back to the pedigree. This is what a pedigree looks like. You can see here, I think this picture has been cut off, but I have another one on this. You can see these are the parents. So when you cross them, you get this kind of siblings. You have like a family tree, okay? And this round blue stands for the wild female, the square stands for the male, Then the one that is mixed, blue and red, stands for heterozygous male. The circle is homozygous, heterozygous female, like that, like that. So you have a pedigree. It is also used by breeders for breeding purposes. And the first year, like I said, any breeding program takes nothing less than 7 to 8 years. So in the case of pedigree breeding too, it takes a while. So the first year hybridization is done. So what you do is you select two pure lines, two parents, and then you form hybrids. That is what is here. This is the parental generation. This is capital S. This is small s. You get a gamete, capital S and small s. This is a hybrid. So you now later go on in the second year. You get the seeds. When you self them, you get the seeds, and you produce F2 seeds. That's you self F1 to get F2 seeds. So from the F2, you select plants. I told you any breeding program is more about selection. You select plants for what you want, whatever traits you want. And you are familiar with this Punnett square by now. Please try and know this square, because like I said, I don't know what is coming out, but I have my question set already. So you should be able to know the possible combinations and the possible gametes that I got from dihybrid crosses. So these letters could change. You may see capital Y, small letter y, any kind of letter. So you should be able to know which is which, so that you are not stereotyped to one particular kind of letter. So this is basically reading. Year 2, F1 generator is the generation itself to produce F2, and you select 15 to 30 plants from F1, and the seeds you collect it also from the F2. In the third year, then you now plant out. You plant it. You plant the F2 population. That's 2,000 to 10,000 plants. And out of that, you select only 100 to 500 out of 2,000 to 10,000 plants. So you have to really be able to select, and then you harvest them separately. That's what is done in the third year. And this is what is done in the fourth year. You self again to get your F3. And from the F3, each progeny should have like 30 or more plants. You harvest them individually, keep records, and then you remove the diseased ones. You keep selecting for the superior ones. Leave the superior ones, remove all the ones that are not bad. You eliminate them. And year 5, you self again. So this breeding program is more about selfing. Okay. So you do selection again as the previous year. You just keep selecting and selfing and selecting and selfing till you get, and you keep records as well. So you get the desired result. Year 6, you do what is called the family rule. That's you self to get F5. And you remember that when you keep selfing like that, you are increasing the homozygosity, right? And you are decreasing the heterozygosity. So you keep eliminating the undesired family, keep eliminating till you get what you want. Then year 7 and 8, you perform what is called a preliminary yield test. Okay, you plant according to your blocks. You plant them as if you are planting experimental plots. So year 7 now you have F6 and F7. So you plant them in blocks like this, okay, for uniformity, so you'll be able to see if everything is uniform like that. So you are planting them in blocks, and then you want to see them clearly, because now the seeds are fewer. You know, you have been doing a lot of selection, removing, eliminating, so the seeds are fewer. You can plant your preliminary yield test. That's what you do in your year 8. And year 9 and 10, you now do what is called multi-locational trial. That's you plant the few seeds you have in different locations so that you'll be able to know the performance. Is the performance the same if I take it to the north, plant it in the north, and plant it in the south? Is it the same or is it different results I will get? So you want to know the yield in different locations. So that is what you do in your year 9 and 10. And of course, in year 11, you have your breeder seed. And what you want to do is that you want to increase them and get a new variety. So you can see in this particular pedigree method, you are even going to use 11 years. So no breeding work is easy. So that is 11 years before you can get a new variety. Now for seed production and distribution, there are different classes of seed. There are 4 recognized classes of seed by the certification agency. We have the breeder seed, the foundation seed, the registered seed, and the certified seed. Those are the 4 classes of seed. For the breeder seed, that's the one we have been doing. All the breeding work you are doing, what you get at the end of the day is the breeder seed. And that one is usually small in quantity. It's not much. Is small in quantity because the breeder must have done a lot of work. So the few seed he has is what is called the breeder seed, and it is usually pure. So you don't see it anyhow. You don't see breeder seed anyhow. They are small in quantity and they are usually pure. So what the breeder does is that they give it to the farmer to form what is called foundation seed. Okay, even this foundation seed is not what you see in the market, but it is more than the breeder seed. When the breeder seed, when they plant it out and it multiplies, then they get foundation seed. So you can see here that the progeny of the breeder seed is the foundation seed, and the genetic identity and purity is still maintained. It is not tampered with. It is just an increased form of the breeder seed. So the identity, the genetic purity is still there. And foundation seed is the source of certified seed and registered seed. So it's the foundation seed now that they will further give to farmers. That's what you buy in the market. Okay, it's the foundation seed you buy in the market, and you want to use it for your own multiplication of seed. So when they now give it to the farmers, they now multiply it further to get registered seed. So registered seed is direct increase from the foundation seed. So you can see how the process is from breeder seed to foundation seed, then to registered seed. And then it now comes to certified seed. Certified seed is the progeny of the foundation seed or registered seed, and that is what you see in the market. Okay, its production is guaranteed by inspection. So as a farmer, when you are working on a very large expanse of land, they give you seed, you plant it, and then these certification officers will come and inspect it just to make sure that the seed is still pure. And you have elements of seed production. Is usually genetically pure, and the seeds will be multiplied on a clean land. Yes, you should have a clean land. Your field is usually very far. It's far inside the bush so that no contamination will come, and it should be in isolation. That's what they call it. You plant in isolation when you want to do seed production, and you have to follow the proper cultural practices. That's fertilizer application, insecticide application, weed control, and so on and so forth. And also field inspection is important. You always have to go to your field all the time. If a breeder selects you to be able to promote a seed, you have to impress the breeder. So you don't have to do things that will make the seed to be contaminated. You visit the farm all the time, then you rogue the diseased plant, remove the diseased plant, varietal mixture, because no matter how careful you are, you still see some off types, that's what they call them, on the field. But they are usually very few. If there are much, then that the whole experiment is gone. The seed is not useful. But they can still be maybe 1 or 2 out of maybe a whole 10 acres. You see maybe like 5, that is not a problem. But if you see them everywhere, then the whole thing is defeated. And once you see off types like that, you remove them. Then harvesting must be done at the right time, and drying and threshing should also be done proper timing. So these are things that I want you to know, okay? And now it's time for revision. So like I said, we have come to the end of this course, and from your course material, let me share your course material. We started with, hope you can all hear me. Can you all hear me?
Yes, ma'am, we can hear you.
All right. Okay, good. So we started in the beginning of this course with definition. We defined crop breeding, we defined gene, we defined allele. For those of you that were not in class then, you are lucky that you have a recording. So I just want us to just quickly move with the with what we have done so far. We defined some terms. Crop breeding, gene, then we talked about biological variations. We talked about the causes of biological variation. I think Farouk was in class that day. We talked about it extensively. Then we moved on to cell division, meiosis, mitosis. We talked about the differences between mitosis and meiosis, gametogenesis, fertilization. We talked about that. I just want to refresh you people's memory. And then we talked about gene symbols, terminology. We talked about definition of gene, characteristics of a gene. Then we went about to talk about the symbols. Okay, we defined phenotype, genotype, homozygous, pure line, heterozygous. We defined all those things like that. So those are things that, like I said, your definition should just be brief. Shouldn't go off line or anything, off points. Just go straight to the point, because the marking guide, you mark points, not story. So I told you that Gregor Mendel is the father of genetics, and we talked about the experiments. So like I said, your dihybrid cross, please go and know it. The monohybrid cross is not difficult. The dihybrid cross, I won't tell you to do a trihybrid because it's quite cumbersome, but you can just know it for your own purpose. But this dihybrid cross, you should be able to know the gametes that are produced to do this Punnett square. If you see AB here, if you see YZ, you should be able to know what to do. So it's not compulsory that it should be AB, and you should be able to know the phenotypic ratio and the genotypic ratio for that. And your chi-square also. Your chi-square is better for you to know it. There are simple calculations that you can know. Your chi-square, your heritability. After chi-square, we talked about heritability. Like I said, you don't need to take any chi-square table to the exam hall. You shouldn't go to the exam hall with any sheet of paper. Anything you need is inside your question paper there. So you know your formula for your chi-square and you work around the examples. Then for your quantitative and qualitative traits, you should be able to know the differences between them. And your heritability, two types, broad sense, narrow sense. You did the calculation also. Those of you that submitted, I marked it. So you should be able to know what to do to calculate your broad sense and your narrow sense heritability. Because there's always one thing about mathematics, it's usually straight to the point. You may not get the answer in the end, but you may not miss everything, because especially if you do the marking, if you go step by step in your mathematics. So it's always easier for you to do questions that have mathematics in it, especially when you know that you know it, other than the ones that you just be writing story and be writing all manner. So also your selection response, like I told you, I don't know which question will come out. So you may see all these, it's not likely you see everything inside one question, but you know, I don't know the way they will do it in DA, but I know that you must come across calculation, and it's not beyond what we have done in class. So your genetic advance should be able to know how to calculate it, especially from your heritability value. So just work around it. And of course, we did kinds of flower, perfect flowering, perfect, monoecious, dioecious, and we talked about features of self-pollination. We also went to talk about self-incompatibility and male sterility. Okay, like I said, it's always a question that I like to set, just to know if you know it or you understand things better. So you should be able to know the types of compatibility and be able to explain them, just short notes, nothing so much. And then, you know, we have covered a lot already. Then I think the next thing here is breeding programs. Steps for controlled crosses and sources of breeding programs. So the steps, okay, the steps that are involved in your breeding program. You have the choice of parents, emasculation, pollination, tagging, and harvesting. You should be able to explain say short things about it. You should be able to say short things about it and say things that are meaningful. So then we went ahead to do method of using pure line selection to improve self-pollinated crops. So that one too is not a big deal. It's not a big deal. You have the first year, the second year, the third year, and the fourth year, then the fifth and to sixth year. All this selection story is not a compulsory that is there, except maybe there's a place where I asked you to define selection, maybe, I don't know. But if you see, if I said, okay, what is the method of using pure line selection to improve self-pollinated crops, you should be able to know what to write there. So then talk about mass selection. That one too, there's no big deal in mass selection as well. And today's class, I talked about pedigree selection and seed production. So I think that is that should be it. So any question for me? Any question for me?
Yes, ma'am, I have a question.
Okay. Yes, that question that you sent to me. Oh, okay. The phenotypic variance was there, right? I think the phenotypic variance was there. You were talking of one past question like that. So if it is there, then there's no need for you to calculate it. But if it's not there, you should be able to calculate it.
All
Yes,
right.
what's your question? I'm listening.
The question I have here is you give us this genetic advance, narrow sense and broad sense heritability calculation. We sent it. You said you would check it and you did not. So
oh, okay. I haven't marked it.
No.
Oh, it's true. It's true. It's true. It's true. It's true. Well, maybe I didn't because I did not see any. Maybe because I didn't see anything strange in it also. Okay, I will look at it, but just know how to calculate it. There's no big deal about it. I will look at it though.
We are just wondering that are we on track or no? That's all.
Yeah, I think so. I'll check it again. I'll check it because I've been busy all the while, but I'll check it again. There's no problem. So I'll check it. But just walk around the questions like that. Just practice and.
Okay, and in the situation where they did not give us any letter, is it not going to be affected when students use different type of letter? Because one of the past question they just asked us to do the hybrid without any letter.
Okay. Well, that one was like that. That was like that. It may not be like that this year.
Okay.
I may decide to give you letter and tell you to work to do the hybrid cross and do your Punnett square. So you should just don't be confused with the letters. That's why I set the question like that year, so you'll be able to use the letter that you're comfortable with. But if you see letters that I said and I said you should use them, then you have to use them. So just practice and know, so that when even if I bring any letter, even if it is Z, you'll be able to know what to do. Okay?
Okay.
Any other question?
No problem.
Yes, ma'am.
Okay.
Please, I have a question.
All right.
My question, how can we transfer the dominant gene to the next generation?
How can you?
Transfer a dominant gene to the next generation.
You should be able to check that one inside your book now. Is inside your course material? Is it me that will be telling you how to? Is it your course material?
I went through it, but they said I don't know.
Open it. Open it in your course material. Let me see.
Gloria, I beg. I beg.
I don't know your course material
anything that they will not ask us, please. That should not bother us at this material time, please.
You know, Gloria is a first class student now. So she wants to know everything.
Okay.
Abby, is you now you want to kill your lecturer? She wants to get 100/100. So give her. Ask your question, Jerry. I don't think you should have a problem, and I don't want to see any of you in this class next session. You should all pass. So I don't want to see anybody's face.
I mean, you will not see anybody. You will not see anybody by God's grace.
I don't want to see anybody's face.
We have another concern. Our exam will start on 25th, which that on 25th might likely.
Good evening, ma.
Might likely be. Sorry, I'm listening to Ma first. I'm coming. That 25th might likely be what?
Public holiday.
If it is public holiday, it's better for you now. They will shift it. You can't do exam on public holiday. It's not possible. If they call something public holiday, then they have to shift it.
Okay.
Yes, please. I'm listening.
I'm sorry, Mama. I don't know if you talk about the previous assignment you gave us that you said we should attempt, but I don't know
Yes,
how to go
yes,
about
that's
it.
what Umari is saying. You know, I just completely forgot about that assignment. But I think it's because I didn't see any problem with it. That's why I didn't point. You know, I'm used to pointing out problems or so. But like I said, I will check. I'll try and do that this weekend by God's grace. Let me just even settle you people and let everything be over with this weekend. So I will answer your
All right.
yes, I will do that this weekend by God's grace. As for
Thank you,
303, you said it's clashing with one of your classes. Was it like that before? Because we have
No, ma.
303.
The lecturer.
So what happened?
Yes, the lecturer last week, he said he won't be available on that
Okay.
on Thursday. That's why he now shifted it to
Okay.
Wednesday. That was what happened.
Okay. Okay. So tomorrow, I think for 303, we still have one more class before revision. We still have one more class before revision because it's like that course is not really. It has a lot of big names, so you people are not really interested in it, but you don't have any choice anyway. But
I'm
I
interested
think
in.
we have a course.
Please, I'm
We
interested
still
in it. That's my.
okay, no problem.
Ma, we
We'll
are
meet
interested,
tomorrow.
ma.
We'll still have a class tomorrow before we have revision. I think, what day did you call your exam for 303? You said? I didn't hear you. Well, 2nd of May.
Yes, ma'am.
Yeah, okay. I think we can still squeeze out one more class. We can still squeeze out one more class before, or whatever. But I know that we won't end the class tomorrow. We won't end the class tomorrow. So please, like I said, if you don't understand anything, you people should consult each other. Try and teach each other, do revision, but I don't think you should have a problem with this course. I don't think so. Everything is quite straightforward and they are just short things that you should know. So, like I said, just try and study hard. And for every other course, I think you people don't have issues too with other courses. Your lecturers are coming, and because we don't want anybody to carry any course over. Anybody, if left to me, if it is only 5 or 6 or I think 7 of you that used to come to class out of the 100 and something that pass, I'm fine with it because there's nothing I can do. I cannot open anybody's head and put book inside. And if you say you should come to class, it's because of things like this. At least there's a difference for you to study yourself and for you to be in class. There's always a difference. So please try and form groups and do revision. Do revision. If you have past questions, do it. If you have things, you do it. So that is the only way you can make progress. And if you still don't understand something, you have already done it among yourselves, you still don't understand, then you can let me know. You can let me know. But I don't think there's any issue. So far, so good. So, any other question? Any other question? I think that is all with your CRP305, crop genetics and breeding. All the definitions, just try and know them. Short definitions, straight to the point. No dilly-dallying, no beating around the bush. We say explain briefly, explain briefly. If we say explain, if you say discuss, you know what to do. If you say state, list, you know what to do. So I don't think you people have any problem. In the absence of anything, I think it's okay to call it a day. Or any question? Gloria,
No
any
question.
question?
Ma.
Okay.
So towards the last topic on the course material, there's some little calculations.
Which one is that? Report material. Which one is that?
Under heterosis of F2 and sub.
Under heterosis. In your course material?
Hello, ma.
Where?
Oh, God. Hello, ma.
Yes, I can hear you.
Okay, under heterosis, ma.
Is it in your course material?
Development of synthetic varieties. Yes, ma.
Development of, let me check that one now. Under
Synthetic varieties.
heterosis. Oh, development. Oh, no. Don't. I didn't. And if you want me to teach you, it's okay. But I don't think this calculation, right? No.
Yes, ma. All right, ma.
I didn't teach you this, so I don't think I have any question on this. But there's really no big deal. It's just trying to tell you the homozygosity and heterozygosity. Like I said, if you keep selfing, the homozygosity will increase and the heterozygosity will decrease. It's just trying to give you the percentage of decrease and the percentage of increase. It decreases by, I think, 25%. So it's, if you keep selfing, is tending towards pure. That is what it means. So, and there's a little calculation there, but that you don't go into that. I don't think I have any question on that. So, okay. Is there any other thing? Is there any other thing? For your dihybrid cross practice, just your mathematics basically, just to
Yes, ma.
on it.
Yes.
Okay.
Ma, I'm sorry for asking too much. Under this stability advancement, there's a table to get our, you said K is constant. So if we're given a value, subsequently we have to, are they going to provide that table for us? Assuming
no, I will provide the value that you need. The K value, the constant, right? Because yes, I don't want you to take any paper to the exam or so I'll put the value, the K value in the question there. So mean the question there.
Are we allowed to? All right, ma. Are we allowed to go with calculator?
You can go with calculator.
Okay.
But not,
no, I don't want embarrassment. That's what
you can go with calculator, but don't go with your phone. Don't allow you to take your phone.
Yes, because when we usually does the computer one, they don't allow calculator.
And there's calculator on the computer. That's why. Just calculator on computer.
Okay.
And
Yes,
it's not
ma'am.
everybody that is aware of that. How many people operate computer? Please,
Ah,
ma.
there's calculator on the computer now. Most people don't know.
And some they don't even know how to get out, or even if I don't even know where to bring it out from. Please, ma, my question is based on our calculation in different value. Based on the approximate, how will you know the correct answer? Like maybe somebody approximate 1.6, another person
Don't worry about that. Just do your approximation. If you want to approximate to anything, feel free. You understand? Most likely, they always tell you that you should do it two places after the decimal. That's your approximation. But if you want to do it anyhow, do it. Just do it the way you know it. In most places, it's usually 2 places after the decimal. Okay.
in our value, please, in our care value, there is constant in our material to be 1.76. So I can ask one question. Okay, in our care value, in our cost material, they put in 1.76, right? So I can ask one question, and then now care value to be 1.40.
They put what? I can't hear you well.
I say in our course material, they came they came across one situation I was solving, and I saw it like 1.47. So if I see that question, what's use the one they gave me, or should I use the one they gave in the course material?
If the one in the course material, you know, I didn't get what you said. Well, your network is very terrible. Did someone get it? Maybe you should tell me. I didn't get what she said. Well, it was just cracking up throughout when you were talking. You said the K value in the course material is what?
1.76.
I will give you K value now. I just answered that question now that I will put the K value there. You don't need to cram any K value to the exam. What about if I don't want you to use 10%? I'll put my K value there. So you work on the K value you see there.
All right.
Okay. In the absence of anything, I think you can call it a day. Tomorrow by 10 a.m. by God's grace, we'll meet for 303. So
by what time, sir? What time?
10 a.m. 10.
All right.
Let's meet 10 o'clock. Yeah.
Glory, if you get me, please, let's meet there, please. Let's do some things now. Sorry.
All right, though. So take care of yourselves. Good night.